let's get started. So I want to first finishing up the lab. I think it's lab 10, right? The PCR lab. So last time, if you remember, what you guys did was, well, virtually, is for this lab we have like let me turn on the laser pointer. You have collected crime scene DNA here, right? And then you also have 4 suspects DNA, A, B, C, D. And virtually, you guys ran a PCR. So today in the lab, if we were doing a face-to-face class time, you would run the reaction that you finished doing in PCR on a gel. So what is a gel? So a gel basically is using a sugar called agarose from seaweed, and when you mix it with buffer and pour it, it's kind of like jello. The texture will be like jello. So then you run the sample from your PCR on a gel like this. So you have your remember the locus we're looking at is BX P007, and this is AL, means this is a marker telling you the different size. So the way this gel work is, if the DNA is small, it will run faster, so it'll be closer to this end of the gel, versus the bigger piece of DNA will run higher up like this. And when we do this gel, we usually apply electricity to it. If you remember, DNA is negatively charged. So we apply a current where it's negative on top and positive on the bottom. So DNA naturally will migrate toward the positive end, okay? So the different line that you see is just telling you different size of the DNA. So for example, this line that you have here match up with the number 2 line here. So this number 2 line is telling you 2 repeats. Remember, we're looking at STR. So how many repeats you're looking at? So this has 2 repeats. So telling you this suspect, the crime scene DNA, one of the allele, one of the paternal or maternal chromosome has 2 repeats. And then on the other one, 2, 3, 4, 5 has 5 repeats. Okay. And you look at these suspect here. Can you kind of tell which one matches the crime scene DNA based on this picture here? Wait, I can't see your comment. Yep, you guys did a good job. B is the suspect. So if you were running this in the classroom like we were supposed to, it'll be something similar. It'll be either A, B, C, or D will match the crime scene DNA like that. So for this lab, you have to remember what we're looking at is the different repeats in the exon, in the introns, right? The anomalous region in the DNA. So we're done with lab 10. That's why lab 10 report is still on Wednesday. Okay, are we clear what is due on Wednesday? This lab here is due on Wednesday. Okay, so let's go on. So we finished lab 10. So let's go on to the rest of the meiosis chapter. Well, is there any question about this lab number 1? Okay, I think we were on here. Okay, so we already last time talked about meiosis 1 and 2, right? Basically, if you look at this picture here, it's just comparing mitosis versus meiosis. So they are number one. So for this chapter, you need to be able to tell me what is the similarity between the two. And of course, the obvious is what are the differences, right? So number one, what is similar is that, let me get my laser pointer out. Number one, they start with the diploid cells. That's what is similar, okay? But remember, the diploid cell here on mitosis is somatic cells, but the diploid cell here in meiosis is germ cell, okay? And the other thing you need to pay attention, well, of course, you already know they all go through prophase or interphase. But remember in prophase 1, that's a huge difference between the two because meiosis, you have synapses, tetra, right? The homologous chromosome get together. Okay, just yeah, you don't do part 2 because we didn't actually run the lab. Okay, so you need to know, Samuel. So prophase, the difference is synapses, tetra, right, crossing over, because you need to remember the goal for meiosis is to do genetic variation. So that's why you have crossing over and stuff like that. And of course, in metaphase, because of the synaptonymal complex and the tetra, they migrate to the middle of the cell together, right? So they have this two files versus one file in mitosis. Okay, and in this case, can you see that in meiosis, the homologous chromosome separate into two different cells? But here, you can tell that in this cell here, the homologous chromosome is still together, right? Now, the other key thing is think about this. Mitosis, you start here, you end up here, and these two cells can go through interphase again and go through this mitosis again, straight down, right? But in meiosis, think about it. When you get all the way down to the 4 sex cells, these sex cells can never go back to the top and go through meiosis again. So that's why mitosis is called cell cycle. It keeps going and going and going, right? Until the telomeres gets too short. But meiosis is not a cycle. It's a one-way street. You just go all the way down, become sex cells, and that's it. These sex cells, sperm or eggs, will combine with the other sex cells. Okay, they don't go back to become germ cells. So you guys need to be able to tell me the difference between mitosis versus meiosis. Now, just for the fun of it, look at this slide. Look at the first cell here. This one. N equal to what? What number? N equal to what number? Good, good. So you guys got it this time, right? For each of these cells, N equal 1. So you have maternal and paternal chromosome, and they're the same type, right? But what is the number of chromosome though? N equal 1. That means there's one type of chromosome. But how many total chromosomes is in this cell? Good. The number of chromosome total is 2 because you have 2 copies of that one type that you have. And can you see that? I'm going to do a quick review. In meiosis here, right, we said that homologous come together. And you see here, you have this one duplicated, and they how the homologous separated into 2 different cells. So at this point here, right, you have you go from a diploid, which is here, homologous together, to a haploid, which is homologous separated. Now, if you look at mitosis here, in this cell, of course, they're diploid because mom and dad is together, right? Even if it's split into daughter cell, like this one here, you can still see that the homologous are still together, except the sister chromatins already separated. Can you see the difference? Okay. So here you have 1 chromosome that is replicated. Here in mitosis, you have 2 chromosomes that is not replicated. So that's where a lot of confusion comes in. If you're just counting the individual strand, you go 1, 2, there's 2 here, and then you also count this 1 and then this 2, then you say they're the same, and then you keep confusing this is a diploid, but it's not. Okay, hopefully this is a quick review for you guys. And this is just a table showing you the differences between the two. Now let's spend about a couple minutes. You think about this question, and then I'm gonna try to explain it to you. Or if you think about it, you have a guess, you can type your answer in the chat box too. Okay, this is a long question, right? Let's start with the first part. You need to explain why this kind of mating, that means you made a 4N watermelon with a 2N. Can you get a 3N? Can you guys figure out how? Let's do this part first. Any volunteers? Come on. People, you just give me a guess. Or do I need to call on somebody? Bingo, in, right on. Let me use that pen to show you what he's saying. So you have 4N, right? What in is saying, this is a somatic 4N. When you split it into in half to become sex cells, right? You have this. So the 4N become 2N, right? And then if you cross this 4N with 2N, and 2N you are familiar with. If somatic cells are 2N, the gametes has to be N, right? So remember, this is sex cells here. This is another sex cells. And when they join together, 2N plus N become what? 3N. So that's what N is saying. So that's right on. Okay. Now the next question is, why can mitosis occur normally in this triploid cells, but meiosis cannot? So watermelon, a lot of you like seedless watermelon, don't you? But how do seedless watermelon reproduce? Have you ever thought about it? So this is what's going on when you do this. You have 4N times 2n, you actually make a seedless watermelon because the 3n watermelon can go through mitosis normally but cannot go through meiosis. Why? Come on, somebody tell me why. Misty, that's a good guess. Think about this. You need to remember the goal of mitosis and meiosis. The goal of mitosis is to make genetically identical cells, right? So for 3n to go through mitosis, what do you get at the end? You will get another 3n because it's supposed to be identical, right? So there's no problem. But for meiosis, what you're trying to do is you're going to divide the chromosome in half. If you divide 3 by half, if you divide 3n by half, you get 1.5n, correct? But you cannot divide chromosome by half. You have to have a whole number. So that tells you this: in order for any organism to go through meiosis, okay, their somatic chromosome has to be an even number. If it's an odd number, they can't really go through meiosis. Does that make sense? Because you're dividing the chromosome in half. Does that make sense? Okay, hopefully you get this. Let me know if you don't keep. Now, for all the process of meiosis, the whole purpose of it is to make, remember, genetically distinct sex cells, right? And the reason is because, well, this is sexual reproduction, and when you combine all the gametes, you get a lot of genetic variation. Versus asexual reproduction, they're just clones from the original, kind of like mitosis. And why is it so important? So sexual reproduction, the one main benefit is to have greater genetic variation. Now here's a scenario that actually happened a few years ago on bananas. So why don't you read about it and tell me what you think? Well, same thing. When you look at bananas, you don't really see seed, do you? So all the bananas that we eat these days are actually the exact clone from each other. Ooh. Come on. Let's do the first question first, right? Can only get some of us, but not all of us. And that's why genetic variation is important because that will make sure the species survive whatever stress it go under, right? Now, the genetic variation I talk about from meiosis is the raw material for natural selection. Remember we talked about this the first day of class, right? So we have all these different genetic combination, and some of those will be better survive or fit, will be better fit to a certain environment. So then those that survive have babies, and the babies will have the set of physical characteristic or genetic combination that allow them to survive. And these genetic variation, remember, is from independent assortment, which is in metaphase 1, and crossing over, which is in prophase 1. Yep, Harley is right too. And I forgot to say, and that's exactly what happened to banana a few years ago. There was a pathogen, I think, I don't remember, it was a fungus or something, wiped all the bananas out in South Africa or South America somewhere. So then all the banana died. Okay, so they have to grow another batch from. So that's why banana a few years ago tastes different than nowadays because it's not the same strain. Okay, if you ever wonder, I mean, I'm one of those person going, why does banana taste so bad these days versus the creamy and sweet one before? These days, it doesn't taste as creamy as before for whatever reason, and now you understand why. Now let's look at some math. When we talk about genetic variation, right, crossing over and the independent assortment. So think about human has 23 different types of chromosome. So if you look at how many different combination of gametes you can have, it's 2 to the 23rd. So in every one of you right now, you have 8.4 million different kinds of sex cells with different combination of genetic materials. Okay. Now this is just one of you. So if you think about your sex game, you need to combine with another person. So it'll be 8.4 million times 8.4. So the babies that come out could have 70.6 times 10 to 12. So you can have a baby every day of your life, and they all still be genetically different. Okay, and that number didn't take into consideration of crossing over. We just look at, the independent assortment part. Yep, C is correct. Okay, next we're going to talk about mistakes in meiosis. Remember last time we talked about mistakes in mitosis, it will cause cancer, right? If your checkpoint fails and all that. Now, what happens if you have mistakes in meiosis? So the mistake in meiosis usually happen when you try to divide up the chromosomes. Okay, so a lot of times it's because of the spindles the microtubule spindles, don't attach to the chromosomes correctly. And that's why you get mistakes in meiosis. Now, there are two, if you remember, there are two during anaphase 1 and anaphase 2, that's when the chromosomes get separated, right? Anaphase 1 is homologous chromosome get separated, and anaphase 2 is sister chromatase. And the mistake in meiosis is called non-disjunction. So let's look at the two different kinds, right? Here. Now, the first one is, what if mistake happened in meiosis 1 during anaphase 1? If you look at this picture here, get my laser. If you look at this picture here, right? Once again, this is n equal 1. You have one type of chromosome, but you have paternal and maternal, right? So during anaphase 1, remember these two guys are supposed to separate into two different cells. But because the spindle didn't attach correctly, both of them actually go to one cell. So this one end up empty with this one type. Okay. So then when these cells divide again, so you get n plus 1, that means you have one extra chromosome in this cell. Now this is assuming all the other chromosomes dividing correctly. This is the only pair that didn't do correctly. Okay, so that's why you have the normal n, but you have one extra chromosome. Now on the other hand, this one will be n minus 1 because you're missing one chromosome. So in the case of n plus 1, you have what we call trisomy. Tri means 3, right? And this is talking about when these gametes, n plus 1, combine with a normal gamete, you end up with 3 copies of that one type of chromosome. And this one is called n minus 1, is monosomy. That means you only have one copy. So what they're saying is, if this sperm or egg combined with a normal sperm or egg, you only get one copy instead of the 2 copies of that type of chromosome. Now, the second type of non-disjunction is occurring during anaphase 2 in meiosis 2. So look at the same example here. So we have the germ cell here with homologs being diploid right together. Now in anaphase 1, the homologs separate, so go from 2n to n, right, into the correct cell. Now, but when it separates, sister chromatase at this time, this side here separate correctly. So you see that is n. But this side, the same thing, spindle didn't attach correctly. So both of these sister chromatids migrate to the same cell, but the other one is empty. So you end up with the same thing, n plus 1 and n minus 1. Yep, the correct answer is B. If you don't know why, just look at this picture here. You have n and then n plus 1, n minus 1, right? Now, this is what I was trying to tell you. If you have n plus 1, right? Now, this is the chromosome that didn't get separated correctly. So this is why this egg has one extra chromosome, n plus 1. And when it combined with a normal sperm, which is n, can you see that this long chromosome end up with 3? Okay. So it's supposed to be 2n. If you do the math, n plus 1 plus n, you get 2n plus 1. So it has a normal 2n on the rest of the chromosome. But for that one type here, they have an extra one. So that's why it's called trisomy, because you have three, right? So non-disjunction here is a new word. Well, no, we saw this word before. So this is just loss of gain of an entire chromosome of that one type. So in this case, this is what it is. For this type of chromosome, the long type, you have an extra one, okay? And in this case, if it's chromosome 21, it could be Down syndrome. So here is another karyotype. If you look at this, chromosome 1 all the way to 22, right? Can you see chromosome 21 here? Can you see there's extra copy? Instead of just mom and dad, you have mom and dad and someone else extra, right? So this is what is called trisomy 21 because it's the number 21 chromosome that get an extra copy, 3 trisomy, okay? And it's quite common. It's about 1 in 750. Now look at this picture. This is another karyotype. Now remember, for each type of chromosome, you have mother and father, but see that they never touch. And I need to keep emphasizing only sister chromatids that are connected at the centromere. But paternal and maternal chromosome, they don't attach at all. Okay? Except, of course, during tetra and synaptic. But regular, if it's not in prophase, they don't really touch or connect to each other. So what's wrong with this picture, people? Can you see? Yep, Harley's right. If you look at chromosome 18 here, you have an extra copy. Okay, so that's what you're looking at. Now, a lot of the chromosome disorder happens in sex chromosomes. Okay, the 3 examples I have is Klinefelter, you have 2X and 1Y, and then trisomy 3X, and then you have Turner syndrome, right? X and 0. So look at these. This is Klinefelter. If you look at this karyotype, you will see that number 1 to 22, they're all normal. But see here, instead of 1 copy of X, it has 2. Now, but this individual is still considered a male because it has Y chromosomes. Yes, Misty, that's why. Remember, centromeres are where sister chromatids attach. So that's why you count individual centromere for the number of chromosomes you have. Okay, if they are on, they have like maternal and paternal, they have their own centromere. They're 2 different chromosomes, right? So Turner syndrome, if you look at this picture here, they have only 1 X. It's considered a female because they don't have any Y. Okay, they usually look pretty normal except a little short. That's about it. But people like that with the abnormality in sex chromosome, they usually cannot have kids and consider sterile. How often does non-disjunction occur? Actually, it's quite often if you think about this. The number is about 10%. Every time, so 1 out of 10, right? But you don't see 10% of babies that are born that are abnormal, right? It's because a lot of the mistakes in meiosis will end up as spontaneous abortion. So women always miscarry. A lot of times it's because the baby genetically is not normal. So that's why you don't see as many abnormal baby being born. Okay, so we're done with meiosis, and I want to talk about the lab for meiosis. But before I go on, and the worksheet for meiosis, is there any questions about meiosis? Okay, so this is the worksheet for today. It will look very similar to the worksheet from last time, but I want to highlight some of these things so you can pay attention. So since we're doing non-disjunction today, right? So it's the same cell that you saw on the first worksheet for meiosis, but we are dealing with non-disjunction right now. So if you look at this, I want you guys to highlight these. So we're looking at non-disjunction in this cell, particularly looking at chromosome number 1 during meiosis 1, okay? So in this case, you're looking at the big chromosome here, right? Chromosome number 1 here. So you want to show me if there's non-disjunction in chromosome number 1, what would it look like? And in particularly in meiosis 1, so we're talking about anaphase 1, right? So in this cell here, metaphase 1, this one will look identical to the last worksheet. You have the two big one line up here next to each other, two small one line up here next to each other. You have your spindles fiber, right? Moving across, attaching to the homologous chromosome. So metaphase 1 will look exactly identical as the last worksheet, but it's the meiosis 1. So at this point, that's at the end of meiosis 1. Since we're dealing with chromosome number 1 that is not divided or that is not distributed normally, so you will have both of the big chromosome number 1 on this cell and only one of the small one on this cell. So if I were you, when I draw this picture here, you have the two big one lined up on top, two small one lined up on the bottom, and the spindles attaching correctly on the small one, but I will not draw a spindle attaching to this one side of the chromosome. So then you remember, oh, this spindle didn't attach to this. That's why both of these guys will move to one side, will move to one cell. Okay, so in this cell, you won't have any big chromosome, but you have one small ones. Does it make sense? And then meiosis 2 go on correctly. You have a single file here, and you split them up. So then you end up with n plus 1, and then you have n minus 1. Does that make sense? Yes, Misty, you can ask. What question do you have? Okay, so the second page of this worksheet is the same thing, except we're looking at non-disjunction of the small chromosome now, chromosome number 2, and we're looking at meiosis 2. So we're talking about anaphase 2 when the sister chromatins didn't separate correctly. Okay, so same thing, metaphase 1, you have the two big one line up on top, two small one line up, but this time the spindles in this cell will attach correctly because nothing is wrong in meiosis 1, right? So you end up with the homolog separating, one big one small one here, one big one small one here. Now during metaphase 2, you have big one here, small one here, but your spindle will not attach correctly. Now since we're looking at chromosome number 2, we're looking at the small one. So if I were you, I would draw the spindle not attaching the small one correctly. So only attaching on that one side, but not attaching on the other side. Okay, so that's why both of the sister chromatids will move to that one cell and it will be absent in the other cell. Okay, so that's how you would do this picture here. Remember, we were focusing on chromosome 2 and meiosis 2. And I think you guys can do the rest. So any questions about this worksheet? Misty, if you have a hard time putting putting the questions in Word, just randomly put some ideas together and eventually it will come out. Okay, let's take a break for 5 minutes and then we come back. I'm going to talk about the lab. Yeah, Jessa, we're gonna do lab 11 after the break, so I'll make sure I answer those questions for you. Okay, Misty, you have with centromere if they both sets are together but not touching. In general, Misty, the way to think about this, if they're sister chromatids, that means if they're identical, then they're attaching a centromere. Any chromosomes that are not identical, then they don't touch at all a centromere. That's how you're going to think about it. Okay, let's go back to the lab. If you have the lab already. So we did part 1 and 2 last time. So what you're supposed to do today is supposed to be part 3. Now remember part 3, when you draw all these cells, right? It's from the first cell that you drew in part 1. So you have to kind of follow through. Whatever you start out in part 1 here, you have to use it in the rest of the parts here, okay? So here when they talk about you need to draw 4 sex cells. Now this one is showing you as sperm again. And they want to look at what if you have non-disjunction in meiosis 2. So remember meiosis 2, when you have non-disjunction, is happening in telophase 2 or anaphase 2. Okay, so it's when sister chromatids not separate correctly. So in that scenario, you're assuming that out of the 4 cells, 2 of them will be correct. So those will be N. So you have N here, N here. And these other 2 cells will be, one of them will be N plus 1. The other one will be N minus 1. Okay, but when you draw it, you need to make sure you draw, what you start with. That's what I'm trying to get. If you start with 1 chromosome, actually you should start with here. You should start with homologous chromosome here. 1 blue, 1 red, and since only one of you guys is doing it, you don't have a table of lab mates, so that you only have 1 type of chromosome in this cell, and you have paternal and maternal not touching, right? And you're looking at G1, so they haven't replicated yet. So paternal red, paternal blue, maternal red here. Okay, one type. And then you go on and on about all of this. So when that first cell that you drew becomes sex cells, right, which is here, you have blue, red, something like that. They all separate into 4 cells. So here is the same thing. When the N is correct, then you look at the top what you have. So the N will be correct with 1 blue, 1 red here. And this one, if it's N plus 1, you have 1 blue and red together. And you have nothing in here with N minus 1. Does that make sense? Now, can you answer question number 2? What percentage of gametes have incorrect chromosome? If these two are N, so what? 50% will have incorrect chromosome number, right? These two. Now, Jess's question is about question 3 to 5. Now, here what is telling you that you're going to draw a normal egg. Okay, so the normal egg will kind of look like your normal sperm up here with the N, right? So you have a blue and a red here for that one type of chromosome. And they want you to join this egg with the abnormal sperm. So you can pick one of these, N plus 1 or N minus 1. If you pick N minus 1, this guy will be empty. If you pick N plus 1 here, you have the blue and the red here. Oh, the egg here, sorry, it'll be just either blue or red. Won't be blue and red together. So this egg should look like one of these sperms here. And this one, let's say if it's N plus 1, you have the blue and red together. If it's N minus 1, you leave empty. So then they want you to draw when you combine these two, what do you get? Do you get 2N plus 1 or 2N minus 1? So you just put all the chromosomes you see in these two cells into one. How come I don't have a question number 5? Am I missing something? So N plus 1 is when you have paternal and maternal chromosome all together in one cell. Remember in meiosis, paternal and maternal are supposed to be separate in 2 different cells. So in this case, N plus 1 is when you have both the blue and the red in the same cell for that one type of chromosome because they didn't separate. You guys have question 5? How come I don't? I can show you another picture just to be sure that you get it. So this is what you're looking at, meiosis 2, right? So you see that, of course, you have 2. So for the 4 sperms that you're supposed to draw, it's supposed to look like this. On part 3, question 1. Right, you have 2 that's normal because you're looking at one type of chromosome. So you guys all have one type of chromosomes, but they're diploid, right? Here, start out in meiosis 1. And then meiosis 2, the sister chromatins then separate, this size separate normally. So you have N, so you have the sister chromatins separated. So in a sense, yeah, Jessa, you're right. So the 2 didn't separate correctly. So you have both of the, let's say this is paternal, move to one cell and the other one doesn't have any. So then the question after that, so the first 4 cells, you're supposed to draw this, right? And then the next one, they're asking, what if you take a normal one combined with an abnormal one? Okay, so you can pick the abnormal one as one of these two, and then combine with a normal one, look like this. This is the N. So if you combine N with N plus 1, these two, can you see that this chromosome here, you have 3 instead of just one of mom and one of dad. You have an extra copy of dad. Yeah, I think that's what Jessa is trying to say. Yeah, so you're correct if you're thinking that way. Yep, Jessa, N minus 1, just like this picture here, has nothing. Can you see that? So Aneri is asking, how would you represent the chromosome number of the zygote? Now, if you're combining a normal gamete with a gamete, let's say you combine these two, that's N plus 1. So if you do your math, N plus 1, you get what? 2N plus 1, correct? So if you're combining situation like this, your zygote will be 2N plus 1. That means all the rest of the chromosome is 2N, but has one extra chromosome in that one type of chromosome here. Okay. On the other hand, if you're combining a normal gamete like this with an abnormal one like this, N minus 1, okay, so then you have same thing, N plus N minus 1. So you end up with 2N minus 1. Can you see that? Oh, that's number 5. For some reason, on my lab menu, I don't see number 5. So that's good. So what else did I not? Let's see. I answered number. I think I answered. Okay. I answered 3 and 4, right? And then you guys asked me about 5. So what is question number 4 then? It's just where you got lost from. We can start from that point. So you see that I was trying to tell you this picture here, the 4 gametes, you're just going to draw it like this picture here. The 4 gametes on the bottom. You have 2 normal, and then 1 that's N plus 1, 1 that's N minus 1. Oh, I'll answer Miss C's question. Thank you. I'll talk about that in a minute. So for this one here, you just copy what this is in the PowerPoint here. Good so far? Okay. So then you have those here. Then you just take a normal one. So let's say these two are normal. So copy that exact picture here. And then for this one, you can pick either the N plus 1 here or the N minus 1 here. Let me show you a picture. So for the egg, you can pick one of these, which is normal. For the sperm, you can either pick N plus 1 or N minus 1. So that's what you're supposed to do here. So you have one chromosome here, you have nothing here, N minus 1, or you have both of the paternal maternal here, which is N plus 1, right? And when you combine to a baby, this is a zygote. If this is N plus 1, then you get 2N plus 1, and that will be a trisomy. Okay, if this is N and this is N minus 1, you have 2N minus 1 for the zygote here, and that's a monosomy. Yes, you can include, so let's start from the top. So you guys have a lot of questions. So start with Aneri's question. Yes, if you start out drawing the key is to follow through. If you start with crossing over, then you want to follow through with crossing over in all of them. But the key point for this part of the lab is not to look at crossing overs, is to show you what happens if you have a mistake in meiosis, how the chromosome number is not going to add up, okay? Yes, that's the whole goal, is trying to show you what happens if you have a normal, just like in a daily life, if a normal person marries somebody that has an abnormal gametes and they come out with a baby that's abnormal. So this is trying to show you how the abnormal baby comes about. How do you get a Down syndrome kids? Because one parent will have normal gametes, the other one will have abnormal. Usually it's n+1. So you end up with chromosome number one having 3 copies, because 2 of those copies will be from paternal or maternal, and then the other one will be from the other one, right? Okay, Misty, if it's monosomy, means that the one of the cell here will be empty, so it'll be n-1, and the other cell is normal, so it's n. So you have n, like you said, plus, no, you have it wrong. So it's n here plus n-1. So you end up here with 2n-1. That's monosomy. Yep. It's 2n-1, right? So monosomy is when you combine a gamete that's n-1 to a normal one, which is n. So that's what you're supposed to draw. Yep, and there is correct. Any questions about this lab? Yes, so a couple of things I want to talk about. So we're still doing study group today at 11:30. The same link that I have up for last time, it still works. So this one is supposed to be my personal chat room. So it always worked for me. So at 11:30, I open up the meeting with this link, so you guys can join and ask questions about all that stuff. Um, Now I want to talk about, if meiosis, you have to really understand meiosis. Because if you don't understand meiosis, you won't be understand genetics. That's coming up right next on Wednesday. We're going to talk about genetics on Wednesday. Okay, so you want to make sure you do all your worksheet, all your lab to get all the steps in meiosis so we can go smoothly in genetics. That's number one. Number two, um, about exam. So all of you supposed to have done the exam already. So what I would do is I would go in and grade the exam, and I always look at every questions. So if the computer grade you wrong, like for example, one of the matching one for cyclin and CDK, the order of how it's activated and stuff, there's actually another way to do that. So I will look into each question to make sure it give you the highest points possible. So you don't have to worry about that. And I will try to get it done by the end of the week so you can see your score. Okay. So from now on, all the exams will look kind of like that. You have half multiple choice, half essays, and you really have to still do notes before class or after class, because they'll help you organize your material. Otherwise, you won't have enough time. Okay, Ian. Yeah, say the question again. I don't see a question from you. Yeah, Annie, will you show me your question again? I can answer it. But this is the link for the study session today, and we are actually done early today. So if you don't have question, you can leave. If you have question, you can stay around. Um, or we can just stop this and wait till study session time. Yes, I'm gonna click this. Last chance, people. Post your questions, or I will see you during um, the study session. Remember using this link. Okay, and I see your question. You said, should I change it to blue before it combined with the egg? Blue. What do you mean change it to blue? Are you talking about? Let me see. I'm following you. Are you talking about the chromosome color? Because what you should do if you're talking about chromosome number, remember from the very first cell here, you should have a blue and a red homologous chromosome, same size, same centromere location, right? And you will follow those chromosomes through all the steps. So here at the end of meiosis one, you have the blue that has replicated with sister chromatids here in this one cell. This one will be red with replicated sister chromatids, so two legs join at the centromere in this cell, right? So the paternal and maternal separated. So that's meiosis one. And then when you draw your sperm, you have a blue here, red here, because the sister chromatid separated. And then when you follow through here, if it's normal, you can draw two blue normal or two red normal, whatever it is. And then for this, you need to follow through, just like what you did in the first one. In this one, you have your blue and red together. You see, I still don't see your first part. Okay, let's do it again. For this one, you have your blue and your blue, and then you will have your red stay together in n plus 1, and then nothing here in n minus 1. No, still don't see your first part of the question. If it still doesn't show up in, why don't you email it to me in D2L, and I'll copy and paste, or I can just say it out loud. Okay, some or all of the sperm DNA is red. Okay, and think about this. I think you know the answer yourself. When does crossover occur? Prophase 1, right? We're already way past prophase 1, so there's no more crossing over. So whatever you end up with after meiosis 1, then you have to use the same color scheme, whatever it is, all the way through meiosis 2, since there's no more crossing over. So you cannot, I hope I'm understanding your question correctly. You cannot change, let's say the sperm DNA is red. You have to keep the sperm DNA. We shouldn't say sperm DNA. We look for each sperm DNA, there's either red or blue DNA, maternal or paternal, right? So after meiosis 1, the paternal will look the way it's supposed to be after meiosis 1 all the way through meiosis 2. So if your sperm has blue DNA, it will stay blue. It won't, even though after it combined with the egg, the blue will stay blue and the red will stay red as two separate chromosomes. They don't combine anymore because there's no more crossing over. Does that make sense? So in a sense, you're keeping track of individual chromosome as it is. There's no more genetic materials recombining or mixing at this point. Not sure if I'm understanding you in, but I hope this is what you're asking. Yes, so for the n plus 1, it will have, in this case, it will have the two sister chromatids, maternal sister chromatids attaching at the centromere. Remember, just like this picture. Let me show you this picture again. See, for the n plus 1, you see this? Let's say this is paternal, right? the sister chromatids didn't separate. So they both go to the one cell here, while the other cell doesn't have anything, right? So when this cell combined with this cell, which is normal, you have a light one and then two dark blue one. So the dark blue one doesn't change to light blue one. So same thing here, right? You have two uh, connected sister chromatids connected at the chromatids here, at the centromere here, with n plus 1, right? So if you draw this same cell here and combine with a normal cell, only have one chromosome, either one of those two, and then you combine, and then you get these two together here, and this one here, just like that picture I showed you in the PowerPoint. Yep, and you're right. So the zygote, the 2n plus 1 zygote, will look like it has two maternal chromosomes, right?